Hi, today we discuss about ultrastructure of nuclear membrane and nuclear pore complex. Structure. The nucleus is the largest cellular organ in animal cell. The nuclear envelope has a complex structure consisting of two nuclear membrane and underlying nuclear lamina. The nucleus is surrounded by a system of two concentric membrane called inner and outer nuclear membranes. Like other cell membrane, each nuclear membrane is a phospholipid bilayer permeable only to small molecules. Outer nuclear membrane are joined at nuclear pore complex through which a small polar molecule and macromolecules pass through the nuclear envelope. Inner surface of inner membrane may be plastrated by a coat of fibrous protein known as fibrous lamina, nuclear lamina or nuclear cortex, and it is formed of a network of intermediate filament called nuclear lamins. Nuclear lamina underlying the inner nuclear membrane, a fibrous network that provides structural support to the nucleus. Nuclear lamina is common in invertebrate cells but absent in plant cells and oocyte. During early mitosis, the lamina proteins get phosphorylated and repolymerized into ilein monomers in telophase. These monomers undergo repolymerization and bind with chromatin for the construction of nucleus. Function of nuclear lamina. Fibrous lamina provide a mechanical support to the inner membrane and also serve as a site for the attachment of chromatin. Inner micelles membrane has a specific proteins which serve as a binding site for the nuclear latins. Nuclear pore complex. Nuclear envelope is a porous covering. It is a porous are not wide open channels since they are occluded with an electron dense material. Fitting into each nuclear pore is a hollow cylinder called annules. A nuclear pore and its annules together form a porous complex. The annules projection slightly inwards into the nucleoplasm and outwards into the cytoplasm. It has a cylindrical walls that enclose a fibrillins or amorphous matrix. The annulus walls is formed of two rings, namely outer cytoplasmic ring and inner nuclear ring. Each ring is formed of 8 tubular and annular protein units, known as annular granules. Each annular granule is anchored to the corresponding nuclear membranes by a transmembrane glycoprotein. Nuclear pores are the sites for the selective bidirectional movement of macromolecules between nucleus and cytoplasm. Still, they are not freely permeable to the free diffusion small molecules. Probably they serve as spacers for maintaining the parallel splicings of the two nuclear membranes. The nuclear envelope is the most case in a barrier to free diffusion. Still, in oocytes, it permits the free molecular exchange between nucleoplasm and cytoplasm.